Hello everyone, myself Varna and today I am here with the topic digestion and absorption of proteins and amino acids. So what are proteins? Proteins, they are made up of amino acid sequence and they are the most abundant and structural functional molecule which is present in us. So proteins they are made up of amino acid sequence, and proteins in a normal adult, it is about 10 to 12 kg weight. About 10 to 12 kg weight of an adult is made up of proteins. Out of that, 50% you can see in the skeletals and in the connective tissue, and 50% will be intracellular. Protein, they do not have any storage form in our body. That is, they are synthesized on daily basis. They are being synthesized. In adults, the rate of synthesis of the protein balances the rate of degradation, and it is called as nitrogen balance. Maintenance of nitrogen balance. The rate of synthesis is balancing with the rate of degradation. So nitrogen balance in detail you will be learning when you take up on nutrition. So nitrogen balance means it is the amount of nitrogen excreted through urine or feces or through sweat. So there will be loss or excretion of nitrogen in the form of urine, feces or in sweat, etc. is equaling with the daily consumed dietary protein. So the intake of protein is balancing with the excretion of the nitrogen in the form of through the form of urine, feces, etc. So intake is equal to excretion or degradation. So that is what is meant by nitrogen balance. They are being balanced. Intake through the dietary proteins is equal to the excretion of this nitrogen through urine, feces or sweat, etc. So that is what is meant by nitrogen balance. So in a normal healthy human being, this nitrogen will be in balance. Nitrogen balance, it is called as maintenance of this nitrogen balance. It will be equal. Intake will be equal to the excretion. Whereas during growth phase or post surgery, during growth stage or during pregnancy and lactation, it is in positive nitrogen balance, which means the intake is more but the degradation of the excretion is less. That is positive nitrogen balance. And during starvation or malnutrition or during fever, all those conditions, it will be negative nitrogen balance, which means the intake will be less but the excretion will be more. But a normal adult, it will be equal. Intake is equal to the excretion. Next, let's move on to the sources of amino acid pool. The dietary proteins is around 70 to 100 gram per day, and body proteins, it is 1 to 2% of the body protein turnover per day, that is around 40 gram per day. A 70 kg man has an average protein turnover rate of 400 gram per day, and same amount is synthesized and same amount is excreted or degraded. That is a nitrogen balance. Then the non-essential amino acids are either derived from the diet or synthesized in the body, and the essential amino acids are obtained from the diet. Even if one of the amino acid is deficient, protein synthesis may not take place. The body amino acid pool is always in dynamic steady state. In adults, the rate of synthesis of the protein balances with the rate of degradation, and it helps in maintaining the nitrogen balance. Nitrogen balance is being maintained. So the non-essential amino acids are synthesized from the essential amino acids. It is synthesized in our body, and essential we have to take it through our diet. Moving on to the digestion of proteins. Dietary proteins are digested by a group of enzymes in the GIT. Daily intake of the protein is around 100 to 200 gram, and proteins get denatured on cooking, so it is easily digested. The dietary proteins are denatured on cooking, therefore it can be easily be digested. All these enzymes are hydrolases in nature. The proteolytic enzymes are secreted as inactive zymogens, and these are synthesized as inactive zymogens, and they are converted into active form in the intestinal lumen. So the proteolytic enzymes, they are divided into two, that is endopeptidases and exopeptidases. Exopeptidases you can give as carboxypeptidase. Under exopeptidase, carboxypeptidase, amino, dipeptidase, tripeptidase, all this comes. All these enzymes are in the form of hydrolases, that is class 3 enzymes, and these enzymes are secreted as inactive zymogens, and they are activated only in the intestinal lumen, and this method helps in the prevent of auto digestion in the secretory acini. So activation takes place in the intestinal lumen, and they are secreted as inactive zymogens. Activation takes place only in the intestinal lumens, and this helps in the prevention of auto digestion in the secretory acini. So these proteolytic enzymes include endopeptidases and exopeptidases. So first let's see what are endopeptidases. Endopeptidases, they act on the peptide bonds inside the protein molecules so that the protein becomes successively smaller and smaller unit. And these endopeptidase unit includes pepsin, trypsin, chymotrypsin and elastase. They act upon the peptide bonds which is inside the protein, and the protein becomes smaller and smaller and smaller units. And examples of endopeptidates, they are pepsin, trypsin, chymotrypsin and elastase. Then exopeptidase, next type of proteolytic enzyme is exopeptidase, which acts upon the peptide bond only on the end region of the chain. It acts upon the end region of the polypeptide chain, and that is divided into two, carboxypeptidase and aminopeptidase. Carboxypeptidase is acting upon the peptide bonds at the carboxy terminal end of the chain, and aminopeptidase is acting upon the peptide bond at the amino terminal end of the chain. Carboxy is acting upon the peptide bond which is in the carboxy terminal, and amino is acting upon the amino terminal. So digestion of the protein in the stomach. The digestion of protein is broken down into amino acids by the enzyme pepsin, which is secreted by the stomach, and trypsin and peptidase in the small intestine. So in gastric digestion, the enzymes involved are pepsin and renin. The enzymes of stomach are pepsin and renin. The gastric HCl provides optimum pH for the action of pepsin and also the activation of pepsin. In stomach, there is hydrochloric acid which is secreted, and it makes an optimum pH for the action of pepsin and also for the activation of pepsin. And the HCl also helps in denaturation of proteins. The acid also denatures the proteins, and it also destroys the microorganisms which is found in the stomach. So gastric digestion, it is by the enzymes of the stomach, pepsin and renin. The gastric HCl provides the optimum pH and also activation of pepsin, and it is also helping in the denaturation of proteins and destroying of microorganisms. So let's see in detail with the pepsin. Pepsin, it is secreted by the chief cells of the stomach in the active form. Inactive form, that is pepsinogen. So it is synthesized in the stomach as inactive form, that is pepsinogen, and it is activated by our HCl. It is activated by the removal of 44 amino acids from the N-terminal end by the HCl, which you see in the gastric acid, and by autocatalysis. So the chief cells of the stomach is secreting pepsinogen, which is the inactive pepsin that is activated by the HCl by removal of 44 amino acids, and also by autocatalysis it is converted to pepsin, and it requires an optimum pH of around 2. Pepsin will be more active at an optimum pH of around 2. Pepsin, it is secreted by the chief cells of the stomach in inactive pepsinogen, and there is conversion of pepsinogen to pepsin, which is brought about by the removal of 44 amino acids from the N-terminal end by the hydrochloric acid, and it requires optimum pH of for its activity around 2. And pepsin is example for an endopeptidase. Pepsin is an endopeptidase which hydrolyzes specific peptide bonds. Pepsin catalyzes the hydrolysis of bond formed by the carboxy group of phenylalanine, tyrosine, tryptophan and methionine. By the action of pepsin, proteins are broken down into proteases and peptones. Proteases and peptones are formed from pepsin. So peptin is splitting the protein to protease and peptones. Next enzyme is renin. Renin, it is different from renine. Renin, 2N is there. Then there is another proteolytic enzyme that is called as renin. Only one N. So renin is the proteolytic enzyme which you see in gastric juice, and renine that you see in kidneys. It is an hypertensive agent. It is a proteolytic enzyme which is secreted by the kidneys, and it is an hypertensive agent which you find in kidneys. So don't confuse with that. There is 2N. Renin, it is active in infants, and it is helping in the curdling of milk. It is causing curdling by the conversion of casein to paracasein. So casein is a milk protein, and it is converted by the renin to paracasein. And this paracasein can easily be digested. So pepsin can convert this paracasein, can digest the paracasein. So renin, it is active only in infants and it is absent in adults. Renin, it is also called as chymosin, is active in infants and is involved in curdling of milk. It is absent in adults. It converts the milk protein casein to paracasein by its action, and it helps in the denaturing of that protein, and this denatured paracasein can be easily digested by the pepsin. So just summarizing the gastric digestion of proteins. So the gastric chief cells, they are synthesizing pepsinogen, which is inactive pepsin, and this pepsinogen is getting converted into pepsin by the hydrochloric acid by removal of 44 amino acids by autoactivation and autocatalysis. It is forming pepsin, and there is denaturation of the proteins. Proteins in the diet is being denatured by the gastric acid, stomach acid, that is HCl, and then this pepsin can hydrolyze these proteins into, the pepsin is converting the protein to proteases and peptones. So smaller fragment is formed. So next, pancreatic digestion of proteins. In pancreatic digestion of proteins, the optimum pH for the activity of the pancreatic enzymes is 8. pH should be around 8, and it is provided by the alkaline bile and pancreatic juice. Optimum pH is 8 for the activity of the pancreatic enzyme, and that is provided by the alkaline bile and pancreatic juices. The pancreatic juice secretion is stimulated by the peptide hormones. The pancreatic juice secretion, that is stimulated by the peptide hormones, namely cholecystokinin and pancreozymin. Cholecystokinin and pancreozymin. Pancreatic juice contains endopeptidases, and endopeptidases includes trypsin, chymotrypsin, elastase and carboxypeptidase, and out of this carboxypeptidase is an exopeptidase. Pancreatic juice contains the most important endopeptidases, namely trypsin, chymotrypsin, elastase and carboxypeptidase. These enzymes are secreted as zymogens. They are secreted as zymogens like trypsinogen, chymotrypsinogen and proelastase. All these enzymes are in the form of zymogens. Trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase. So pancreatic acinar cells are prevented from autolysis. They are in the form of inactive zymogens, which protects the pancreatic acinar cells from autolysis of these pancreatic juices. All these enzymes, they have serine proteases. All these enzymes has serine proteases. That is, the active center of these enzymes has serine residues. All these trypsin, chymotrypsin, elastase, they all are serine proteases enzyme, and which means their active centers is serine residues. In case of acute pancreatitis, what is acute pancreatitis? It is a fatal condition where the premature activation of trypsinogen results in the auto-digestion of pancreatic cells. So we were said in normal conditions, it is said that this trypsin, it is synthesized as the inactive form as trypsinogen. So that it is preventing the auto-digestion or autolysis of the pancreatic acinar cells. So in acute pancreatitis, it is a fatal condition, it is a clinical condition where there is premature activation. It is activated early, trypsinogen, and this trypsinogen is getting converted into trypsin at an early stage, and it is causing the auto-digestion of these pancreatic cells. So it is an abnormal condition where this trypsinogen is prematurely activated and forming trypsin, and it is causing the auto-digestion of the pancreatic cells. So that condition is called as acute pancreatitis. So let's see each of these enzymes in detail. First one is trypsin. Trypsinogen is activated by enteropeptidase, also called as enterokinase, secreted by the duodenal epithelial cells, and it is present in the intestinal microvilli membrane. Trypsinogen is activated by enterokinase, present in the intestinal microvilli membrane. Once activated, the trypsin activates other enzyme molecules. Trypsin is activated by the removal of hexapeptide from the N-terminal. Trypsin is activated by the removal of hexapeptide from the N-terminal end of the trypsinogen. Trypsin catalyzes the hydrolysis of the peptide bonds formed by the carboxy group of arginine and lysine. Trypsin it is catalyzing the hydrolysis of the bond which is formed by the peptide bond which is formed by the carboxy group of the amino acids like arginine and lysine. So where is trypsin acting? It is acting on the carboxy group, the peptide bond which is formed by the carboxy group of arginine and lysine. It is synthesized by the is activated by the enterokinase, and it is activated by the removal of hexapeptide from the N-terminal end of the trypsinogen. So the intestinal microvilli is synthesizing trypsinogen. Intestinal microvilli, mucosal epithelial cells, that is synthesizing enteropeptidase, trypsinogen, and trypsin is being activated. Trypsinogen is getting converted into trypsin. Next one is chymotrypsin. Trypsin will act on chymotrypsin, and 3 peptides, namely A, B, C, are being formed, and these 3 segments approximated to form the active centers. Trypsin will act upon the chymotrypsin in such a manner that it is producing A, B, C peptides. These three segments are approximated so that the active site is formed. Thus, selective proteolysis produces the catalytic sites. Next one is carboxypeptidases. Carboxypeptidases, they trypsin and chymotrypsin degrades the proteins into smaller peptides. Trypsin and chymotrypsin together, they are acting and forming the degradation of proteins into small peptides. Thus, a further hydrolysis into dipeptides and tripeptides by the carboxypeptidase is present in the pancreatic juice. The procarboxypeptidase is activated by the trypsin. So, here also, like in chymotrypsin, the trypsin is activating the procarboxypeptidase to carboxypeptidase, and they are metalloenzymes which is requiring zinc as the They are metalloenzymes, and this is being activated. The procarboxypeptidase is being activated by trypsin as carboxypeptidase, and this carboxypeptidase hydrolyzes peptides formed by the trypsin and the chymotrypsin into dipeptide and tripeptides. So, it is acting upon the smaller peptides which is formed by the trypsin and chymotrypsin, and it is further hydrolyzing these peptides into dipeptides and tripeptides. So, trypsinogen is being synthesized, that is getting converted into trypsin, and trypsin is helping in the activation of chymotrypsin, and also procarboxypeptidase is getting converted into carboxypeptidase, and all this is acted, the products formed by the trypsin and chymotrypsin, that is being acted by the carboxypeptidase, and they are forming di and tripeptides. So, initially trypsin is being formed by the trypsinogen, and trypsin is activating the chymotrypsinogen and procarboxypeptidase, and it is converting it into its active form, that is chymotrypsin and carboxytrypsin, and this trypsin and chymotrypsin is forming small peptide bonds, and this small peptide is being is acted by the carboxypeptidase, and this carboxypeptidase is converting that into dipeptides and tripeptides. Next, moving on to intestinal digestion of proteins. There is complete digestion of proteins to amino acids by the succus entericus, that is the enzyme contained in the intestinal juice. Luminal surface of the intestinal epithelial cells contain leucine aminopeptidase, which releases N-terminal basic amino acids and glycine, and there is protein aminopeptidase, which releases proline from polypeptide ends. And the third enzyme is dipeptidase and tripeptidase enzymes, which complete the digestion of proteins to amino acids. So, when coming into the intestinal digestion of proteins, complete digestion of the small peptides to the level of amino acid is brought about by the enzymes which is present in the intestinal cells. So, as the pancreatic digestion, we are getting small peptides, di and tripeptides, that is formed. Further, it has to be converted into amino acids. So, complete digestion of the peptides is taking place in the intestine by the intestinal enzymes which is present in the intestinal juices, that is succus entericus. The luminal surface of the intestinal epithelial cells contains the enzymes, like one is leucine aminopeptidase, which is helping in the release of N-terminal basic amino acids and glycine. Next enzyme is proline aminopeptidase, which removes the proline from the end of the polypeptides, and there is di and tripeptidases, which is bringing about the complete digestion of the proteins. The peptide chains are being broken, and there is complete digestion of that protein. So we are getting the amino acids. So this is the summary of the digestion of proteins. So from the gastric secretion, free amino acids from the gastric secretion is reaching, and there is pancreatic digestion by the trypsin and chymotrypsin and carboxy peptidase. Trypsin is formed from trypsinogen by our HCl. It has been activated, and trypsin is further activating chymotrypsinogen, and carboxy peptidase is being formed by trypsin, and the trypsin and chymotrypsin, they will synthesize smaller peptides. Proteins are getting degraded by the trypsin and chymotrypsin into small peptides, and this carboxy peptidase is converting this smaller peptides into di and tripeptides. Then when coming into the intestine, this is again completely digested into amino acids, and there are certain different types of enzymes, that is, which is acting on the particular terminals or end of the polypeptide, and finally it is helping in the release of amino acids. Next, moving on to the absorption. So protein is being digested, and finally we are getting the amino acids. So this amino acid absorption site is small intestine, and this absorption of these amino acids is an energy requiring process, and there is carrier mediated transport, and there are ATPs which is sodium dependent. The absorption of amino acids occurs mainly in the small intestine, and it is energy requiring process, and these transport systems are carrier mediated or ATP or sodium dependent symport systems. There are about 5 carriers of amino acids. First one is for the neutral amino acids, carriers for the neutral amino acids like alanine, valine, leucine, methionine, phenylalanine, tyrosine, isoleucine, etc. So neutral amino acid carriers. Then there is carriers for the basic amino acids, lysine, arginine and cysteine, and carriers for amino acids and glycine. There are carriers for acidic amino acids. There are carriers for beta amino acids. So there are specifically 5 types of carriers for amino acids. So out of which neutral amino acids, transport of this neutral amino acid is really important. It is by the Meester cycle, gamma glutamyl cycle. Meester cycle, absorption of neutral amino acids in intestine, kidney, tubules and brain is by this method. So we have specifically 5 different carrier proteins for amino acids, out of which neutral amino acids, they are being transported by the Meester cycle, that is gamma glutamyl cycle, is used for the absorption of neutral amino acids in kidney, intestine and brain. The main role is played by the tripeptide and glutamine, glutathione, that is GSH. Glutathione reacts with amino acids to form glutamyl amino acids by the enzyme gamma glutamyl transferase. It then cleaves to give amino acids. So once it enters into the cell, it cleaves back and gives the amino acids. So glutathione is reacting initially with the amino acid and is forming glutamyl amino acid, and the enzyme helping is gamma glutamyl transferase, and then it is cleaving back to give out the amino acids. So the glutathione reacts with amino acids to form gamma glutamyl amino acid, and this is catalyzed by the enzyme gamma glutamyl transferase, The glutamyl amino acid is then cleaved to give free amino acids. The net result is the transfer of amino acid across the membrane. So outside it is combining, once it gets inside it is cleaving to give free amino acids. The transport of one molecule of amino acid and regeneration of GSH requires 3 molecules of ATP. So this is the Meester cycle. So outside the cells is our amino acids that is getting combined with the glutathione and it is forming glutamyl amino acid. The enzyme is transferase enzyme, gamma glutamyl transferase enzyme. It is helping the formation of glutamyl amino acid by combining amino acid outside the membrane with glutathione, and once it is inside it is giving out the amino acids, it is cleaving out the amino acid, and here we get the free amino acids. Then it is getting converted into oxoproline and then glutamate and then to glutamyl cysteine and finally to glutathione. So there is resynthesis of this glutathione, and as a result the amino acid is being taken inside the cell. So in the first step, glutathione reacting with the amino acid and forming glutamyl amino acid, enzyme is gamma glutamyl transferase, and in the next step, second step, it is releasing amino acid and the enzyme is gamma glutamyl cyclotransferase enzyme, where it is releasing free amino acids into the cell. Then it is converting, after releasing it is getting converted into oxoproline, and then this oxoproline is getting converted into glutamate. It is getting converted into glutamate and the enzyme is 5-oxoprolinase, and then this glutamate is getting converted into glutamyl cysteine. It is getting converted into glutamyl cysteine, and the enzyme is gamma glutamyl cysteine synthase. Gamma glutamyl cysteine synthase is the enzyme which is helping in the conversion of glutamate to glutamyl cysteine. Then there is regeneration of this glutathione from this cysteine. Glutamyl cysteine is getting converted to glutathione and the enzyme is glutathione synthase enzyme. And you can see the ATPs is being used in all these 3 steps. Last 3 steps there is utilization of ATP. So there is utilization of 3 molecules of ATP. Third, fourth and the fifth step. Last 3 steps where there is utilization of ATP. So that is with the Meester cycle for the absorption of neutral amino acids. So the summary of protein digestion and absorption. In the lumen, the proteins are getting converted into amino acids, dipeptides and tripeptides, and it is with the help of the gastric and pancreatic enzymes. Then endopeptidase, exopeptidase, they are all being synthesized, and they are synthesized in the inactive zymogen forms, and they are getting converted into the active forms. And trypsin is synthesized as trypsinogen, and trypsin is activating the other polypeptide carboxy enzymes, carboxypeptidase and chymotrypsin. Then at the brush border there is oligopeptides getting converted to aminopeptides and di and tripeptides via peptidase enzymes, and also there is cycloplasmic peptidases involved. Next, moving on to the food allergies. What is meant by food allergies? Di and tripeptides, they enter the brush border of the intestinal epithelial cells and hydrolyze to amino acid and transported to the portal vein. That is what is happening. Di and tripeptides is entering the brush border of the intestinal epithelial cells and they are hydrolyzed into amino acid transported to portal vein. Rarely, large peptides may be absorbed intact either transcellularly or pass in between the epithelial cell or paracellularly and enters the circulation. These produces antibody reactions and allergies. So, the di and tripeptides are the only one which is being absorbed. So, rarely there is large peptides being absorbed, and this can lead to allergy because there is large peptides being absorbed and they are passing into the epithelial cells and they enters into the circulation, and this large peptide is considered as a foreign body, and there is production of antibody against that and it is forming allergy. So, coming to the clinical application. First one is oxoprolinuria. Oxoprolinuria, that is 5-oxoprolinase enzyme is defect. 5-oxoprolinase enzyme that you are seeing in Mr. cycle, that is oxoproline getting converted into glutamate. In that step, it is with the help of oxoprolinase enzyme. So, that is defect and leading to oxoprolinuria. Next one is allergy to milk and fish, that is due to the absorption of partially digested protein. Partially digested protein are being absorbed. Partially digested milk or the fat proteins are being absorbed. Next one is defect in intestinal transport system. There is defective intestinal transport system. Under that comes Hartnup disease, immunoglycinuria, cysteinurias, lysinuric protein intolerance. Hartnup disease is disease you will be studying in the upcoming classes, that is in aromatic amino acids. Hartnup disease is the absorption of aromatic amino acids from the intestine, then from the reabsorption from the renal tubules, all will be defective. Absorption and reabsorption of aromatic amino acids is defective. So, the tryptophan deficiency and it is causing the tryptophan deficiency, and there will be pellagra like symptoms. Next one is partial gastrectomy, pancreatitis, carcinoma of the pancreas, cystic fibrosis. These are all due to the defect. This is all leading to the defective digestion and absorption of proteins. Partial removal of gastric system, gastrectomy. Then pancreatitis, that is premature maturation of the tryps. Then carcinoma and cystic fibrosis is all affecting the digestion and absorption of proteins. Then fifth one is protein losing enteropathy, that is excessive loss of protein through the GI system. Next one, intracellular degradation of protein. Proteins are degraded constantly. In intracellular, there is constant degradation of proteins. Half-life of the protein is known as the time taken to reduce the quantity into its half. Is the time taken to for the reduction of the quantity of the half of the initial value. So, how much time is taken for the protein to make the quantity into half. Half-life of the protein is usually few hours, and there are enzymes which is having only half-life few minutes. Then there is a PEST sequence in proteins, proline, serine and threonine. Determines the ease with which they are being digested. The PEST sequence in protein. This is areas which is rich in proline, glutamate, serine and threonine on a protein, which gives the inherent message to break down the protein, that protein into very quickly. Extracellular particles or proteins are taken up by the endocytes and are fused with the lysosomes. Intracellular proteins are also ultimately broken down into lysosomes. The PEST sequence, they are containing proline, then glutamate, serine and threonine, and on these proteins they give the inherent message to break down the proteins very quickly. Determines the ease with which the protein has to be digested. Extracellular proteins are taken up by the endocytosis and fused with lysosomes and it is degraded. Next, moving to cathepsins. Cathepsins are intracellular proteases in phagolysosomes. Particles are broken down by the enzyme cathepsins. Cathepsins helps in means it to digest. In phagolysosomes, the particles are broken down by the enzyme called as cathepsins. So they are seen in phagolysosomes, which helps in the breaking down of the particles. Cathepsins means to digest. Cathepsins, there are about 18 types. In lysosomes, the proteins are degraded by cathepsins, and there are 18 types, and they are designated from A to T. They are named from A to T, and it requires an optimum pH. It is mostly active at a pH around 3 to 5. Next one is ubiquitin. Ubiquitin, it is intracellular protein breakdown independent of lysosomes. Intracellular protein breakdown which occurs independent of lysosomes. This involves ubiquitin. It is so named because it is seen in all cells abundantly. It is seen in all cells abundantly. It is a small protein with 76 residues. The proteins are marked for degradation by covalent attachment of ubiquitin. It is present in all cells and it is having 76 amino acid residues. It is attached to the protein with the help of 3 enzymes, that is enzyme 1, that is the activating enzyme, then enzyme 2 is the ligase enzyme, and enzyme 3 is the transferase enzyme. This is a non-lysosomal ATP dependent process and involves several enzymes and proteins. The ubiquitin proteasomes pathway. The process is also linked with cell cycle and epigenic modifications. Ubiquitin is attached to proteins with the help of 3 enzymes, that is the enzyme 1, enzyme 2 and enzyme 3, which is activating, ligase and transferase enzymes respectively. Then due to the enzyme 3 defect, enzyme 3, that is the transferase, it can lead to Angelman syndrome or von Hippel-Lindau syndrome. The Angelman syndrome, it is a genetic disorder with an with a gene located. Angelman syndrome, it is a genetic disorder, and the gene located on the chromosome 15 is called as ubiquitin protein ligase enzyme, that is E3A gene. It causes developmental delays like crawling between the age of 6 to 12 months in babies. Then ataxic movement, ataxic movements, there will be no control of the muscular movements. Muscular contraction will not be in control. It can lead to speech delay, etc. Then happy and excitable personality, small sized head and seizures. Next one is von Hippel-Lindau syndrome. It can lead to hemangioblastoma, that is the cancer tumors which you see in retina, brain, spinal cord, etc. Hemangioblastoma, then renal cell carcinoma, pancreatic cyst will all be seen in that syndrome. Okay. Next, moving on to proteasomes. Proteasomes, those are proteins attached to ubiquitin is degraded by the proteases of the cells. Ubiquitin attached proteins are immediately broken down inside the proteasomes of the cells. The proteasome assembles has a central cylindrical hollow core. It has got a central cylindrical hollow core. Ubiquitin attached proteins are immediately broken down and taken inside the proteasomes. Proteins attached to ubiquitin and is degraded by the proteasomes of the cells. Proteolytic enzymes present in proteasomes will digest the proteins into small peptides, which is containing around 5 to 6 amino acids. Next is transport of amino acids in the body. The plasma level of these of amino acids in blood, plasma level in blood is around 1 milligram per dl, except glutamic acid and glutamine level of which is around 10 milligram per dl, except for glutamine and glutamic acid and glutamine, it is 1 milligram, and for glutamine and glutamic acid it is 10 milligram per dl. That is the blood plasma level. Breakdown of muscle proteins is a source of amino acids. Amino acid source in blood is the breakdown of muscle proteins, and liver is a site of disposal of almost all proteins. So source is breakdown of muscle proteins, and it is disposed through the liver through urea cycle, disposal of ammonia through urea cycle. So during fasting condition, the muscles will release alanine, glutamine into our body. In fasting condition, the muscle releases the amino acids, mainly alanine and glutamine, of which alanine is taken up by the liver and glutamine by the kidney. So during fasting condition, the muscle is releasing alanine and glutamine and branched chain amino acid, and alanine is taken up by the liver and glutamine by the kidney, The liver removes the amino acids and convert into urea. The liver is taking the amino acid, and it is the disposal of ammonia. Amino group is converted into urea cycle. And the carbon skeletons is used for gluconeogenesis. The carbon skeleton remaining is being used by the for the gluconeogenesis. Branched chain amino acids valine is taken up by the brain. In fed condition, amino acid absorbed from the diet are taken up by the different tissues. Amino acids in the diet, during the well-fed condition, from the diet it is taken up by almost all tissues. Muscle and brain take up the branched chain amino acids and releases glutamine and alanine. Alanine from the diet, it is being passed to the liver and to the muscles. Both muscles and brain take up branched chain amino acid and releases glutamine and alanine. Muscles give up glutamine and alanine. Alanine is taken up by the liver and then to the muscles. The glutamine is derived to the kidney and aid to the regulation of acid-base balance, while alanine is taken up by the liver, and valine is taken up by the brain. Glutamine is taken up by the kidney for acid-base balance. Alanine taken up by the liver for muscles and also muscles. Alanine to the liver and also to the muscles, whereas valine to the brain and glutamine by the kidney for acid-base balance. Alanine stored in muscles is released into liver back during starvation. Alanine, which is stored in the muscles, is released to the liver when there is starvation. During starvation, the muscle is releasing the stored alanine towards the liver. Thank you.